here at Penn, the Department of, here we go, Department of Psychiatry, Neuroscience, and Anesthesiology. So it's my great pleasure to welcome you all today to the doctoral dissertation defense of Lindsay Louise Edjo, also known as at neuro_melody. So before we begin today, just want to acknowledge the amazing turnout today. So especially want to welcome Lindsay's family and friends, mentors, all the people online. It's really fantastic that you were able to join us today. And so it's really clear a PhD, it's never earned alone. It's really through all of the encouragement, sacrifices, and support played by all of you here today. So thank you personally as Lindsay's mentor for supporting her along the way. It's been fantastic. And so, yeah, we're really glad that we can all celebrate this day together. So Lindsay began her scientific career at the University of Pittsburgh. She earned her bachelor's degree in neuroscience and music, while also making significant contributions in the lab of Brian Davis, and was able to co-author several publications in gastroenterology as well as pain, which is the flagship journal of our field. She then joined UPenn here through the Neuroscience Graduate Group, NGG, where she completed her doctoral work in our lab on what you'll hear today on the medial amygdala and its role in pain and pain relief. And so throughout grad school, Lindsay really has distinguished herself as an exceptionally creative, rigorous, impactful scientist. Her work has contributed to publications in leading journals, including Cell through a rotation project with Dr. Ismail Abdus Sabor, as well as contributing to multiple papers in the lab that have been in Science Advances, in Nature, and elsewhere. And so it really just speaks to her collaborative nature and how her work really has impacted how we think about pain encoding in the brain, and how the role of these like affective, unpleasant experiences can be really leveraged to create new types of translational therapies that one day hope will help patients. So her accomplishments have also been recognized nationally. Really want to highlight that Lindsay received one of the most prestigious awards from the NIH. This is called a DSPAN F99K00 award. This is a very competitive training award. It was actually the first award of this kind awarded to anyone at the University of Pennsylvania. So congratulations to that. What this award is, the F99 portion was for completion of her doctoral work here, and then she'll go on and transfer this into a K00 phase where she's going to be continuing her work down at UT Dallas in the lab of Ben Kolber and Kate Sadler, and I believe I see some names on the Zoom call, so hi Ben and Kate. And then following Lindsay's defense today, again, we want to make sure that we have everyone come join us over in Barchi Library. So after this public defense portion, there will be about an hour-long private defense with Lindsay's committee. And so we ask that you please come join us over there. Lastly, I really do want to also note that, Lindsay's work does not just kind of end at the lab bench. I do want to also acknowledge that she really has become an outstanding sort of ambassador for neuroscience broadly, and that she's really deeply committed to science communication, and also received this other amazing recognition from the National Academy of Science, Engineering, and Medicine, the Eric and Wendy Schmidt Award for Excellence in Science Communication, which was absolutely a huge honor. So congratulations. And so to keep with my allotted time, because we got to make sure we're hitting all these different marks today, I just want to acknowledge that this will represent years of the culmination of hard work, perseverance, and scientific excellence that Lindsay has brought to this very important topic. And so please welcome Lindsay to the podium, and let's do this.
Thank you so much, Greg. Can everyone hear me? Thank you so much, Greg, for the amazing introduction. And thank you all for being here, those in person and those on Zoom. I really appreciate it. And I'm happy you're here to listen to more of the things I've been doing during my PhD. My project entitled Opioidergic Organization of the Nociceptive Medial Thalamus, or you can say Organization of Opioid Responsive Cells, a brain area that processes pain called the medial thalamus. So as many of you know, I've been studying pain for my PhD, which is me that, which is defined as a unpleasant sensory and emotional experience associated with tissue damage. So pain can be acute and be short term. This can come from like stubbing your toe or even a surgical incision. But many times pain can persist and become chronic, lasting over 3 months and disrupting daily activities. In the US, as of 2023, 24.3% of adults were living with chronic pain, with 8.5% or 1 in 11 having high impact chronic pain, which frequently limits daily work and life activities. So, pain is this multi-dimensional experience, right? Imagine you're stubbing your toe. Not only are you processing the sensory component of it, so like where exactly in your toe was stubbed, how intense is that pain, and then what is the quality of it? Is it sharp? Is it dull? But you're also processing the unpleasantness associated with that pain. So you stub your toe and you're angry at the table and you're angry at yourself, and you have this the drive to escape this unpleasant state. So interestingly, opioid drugs can diminish this affective, unpleasant component of pain, though unfortunately hold very high abuse and overdose potential. In fact, right now we're facing an opioid abuse epidemic in our country, in which there are around 200 people passing away every day of fatal opioid overdose. And I'm giving this talk from Philadelphia, Pennsylvania, in which this is a top 10 ranked among all the major US cities in fatal opioid overdoses every year. Over 800 Philadelphians are dying yearly from opioid overdose. And a recent survey says that 1 in 3 Philadelphians personally know somebody who has passed away of a fatal opioid overdose, personally. So pain can be treated with opioids, and for some time, this was the gold standard. But there's a clear and urgent need for new strategies to manage pain that do not promote a transition to addiction. Unfortunately, the road to developing these non-addictive painkillers remains unclear, but our lab's approach, the Quarter Lab at UPenn, is to identify basic science principles of how the brain encodes the experience of pain in order to develop translational targets for non-addictive painkillers. And we do this by identifying pain circuits that express opioid peptides and receptors outside of these traditional reward and pleasure pathways. So one such opioid system we focus on in the lab is the mu-opioid system, characterized by the expression of the mu-opioid receptor. This is the star of the day. And this is one of 4 opioid-binding G protein coupled receptors. The mu-opioid receptor binds both endogenous ligands, like endorphins, which you might have heard of, or enkephalins, as well as exogenous opioid drugs like morphine, fentanyl, and heroin. So the opioid receptor is specifically interesting to us because it mediates the analgesic and rewarding effects of opioids by inhibiting pain neurons. So when an opioid ligand binds this mu-opioid receptor, that causes the cell to hyperpolarize and inhibit its activity. And if you inhibit the activity of a cell that's necessary for processing pain, that can provide pain relief or analgesia. So we want to identify specific pain circuits in the brain that express the mu-opioid receptor. Well, one of the regions most densely expressing the mu-opioid receptor in our brains is an area called the medial thalamus, which you can see here in the stain of the mu-opioid receptor protein in the medial thalamus of a mouse that I did in the lab. It takes on this cool Batman shape, or you could call it an anchor shape. That's actually what first popped out to us about this region. But the first question is, how does the pain that we feel from our internal or external environment, like stubbing your toe or even a tummy ache, even reach the medial thalamus? Let's walk you through the canonical pain circuit. So during a painful stimulus, again, let's imagine you stub your toe, pain signals travel from the foot to the spinal cord, and then up something we call the spinal thalamic tract, sending collaterals to areas like the reticular nucleus and the medulla, before heading to this area called the thalamus. then heading up to the cortex for further processing. So let's focus on the thalamus here. Here are images of the thalamus of a human and a mouse. The thalamus is traditionally thought of this as this relay center for the cortex, gating sensory processing and helping tell the cortex what is happening in our environment, but it also does quite a bit of integration of inputs from subcortical structures to help keep the cortex updated about incoming sensory information. The thalamus is split into the lateral and medial portions, where the sensory discriminative component of pain heads up the lateral thalamus and to somatosensory cortices, while this affective motivational pain unpleasantness part heads to the medial thalamus before going to anterior cingulate and insular limbic cortices and the motor cortex. The medial thalamus is a really important step in this relay of affective motivational pain information, but it's also really involved in things like cognition, attention, arousal, and this affective motivational component of pain. But it's interesting because the role of the medial thalamus in pain has not been a huge focus in the field, especially compared to the lateral nuclei, despite its dense expression of the mu-opioid receptor. The medial thalamus itself is this very highly segmented structure. It's located along the midline and it's divided into 10 distinct subnuclei, all listed here. Now, in this talk, I'm not going to get into the weeds of each individual subregion and how each one contributes in the context of behavior and perception, but I will tell you that many of them have been shown to activate during pain. And they activate to multiple types or modalities of nociception or pain. Here in this study, we're looking at the CM, central medial thalamus. And this group measured population activity, some calcium activity in vivo using a molecule called GCAMP, which is a way to measure neuronal activity by detecting fluorescent changes that occur when calcium binds within the cell, and because calcium levels in the cell rise during neuronal firing. And so this group looked at population activity in the CM and found that the CM increases in activity in response to heat pain, in this case in the form of an immersion of the tail into 52 degrees Celsius water. This other group looked at some calcium activity in vivo as well and found that the paraventricular nucleus increases in activity in response to colorectal distension, which is a form of gut pain. Other groups looked at electrical activity in the form of single unit recordings in vitro and found that, for example, in the central lateral thalamus, this group activates in response to a tap, a hair, a pinch, or an inflammatory bradykinin injection. And then this other group looked at the MD thalamus, other single unit electrical activity recordings, and found increases during a spinal cord injury in rats. So multiple medial thalamic nuclei are activating in response to nociceptive stimuli. Also, mu-opioid receptor agonist inhibition of medial thalamic cells reduces pain responses. So this group here looked at the central lateral thalamus and found that when you bilaterally microinject Damgo, which is a mu-opioid agonist, that inhibits the neuronal activity of these more positive cells, mu-opioid receptor positive cells, increasing the threshold for these rats to emit a negative affective ultrasonic vocalization in response to a tail shock. So in this case, inhibiting this population increases pain thresholds or decreases the pain in these rats, pain responses at least, affective motivational ones. Additionally, something really interesting is the human lesion data between the medial and lateral thalamus. So lesions to lateral thalamic nuclei, which are these sensory discriminative processing ones, actually causes chronic pain and a condition called thalamic pain syndrome, which is a type of central post-stroke pain. But clinical lesions to medial nuclei, like the central lateral thalamus, can actually help to relieve pain. So in this study here, we have lesions to the posterior central lateral thalamus. This was a 96 patient study, and they found that 20% of patients had complete pain relief. There are also many examples of other studies showing that stereotactic lesions to medial thalamic nuclei, like the CM or the Cl or a multiple nuclei at once can help relieve pain symptoms in things like cancer pain, trigeminal pain, or even pain syndromes that come from the lesions to the lateral nuclei. So it looks like a lot of nociceptive stimuli, our pathways, are moving through different nuclei within the medial thalamus, and people have studied different subregions one at a time or even several at a time, but it's unclear how each one contributes to this experience of pain and responds to pain comparatively, leaving many open questions about this population, including, is nociceptive stimulation uniform across medial thalamic nuclei? What's the differences between acute versus chronic nociceptive processing in the medial thalamus? Are mu opioid receptors expressed directly on these pain-active nociceptive medial thalamic neurons? So would mu opioid ligands be able to directly inhibit these nociceptive cells in the medial thalamus? What is the circuit architecture of medial thalamic moor neurons? So who in the brain, what brain regions talk to the medial thalamic moor cells, and then in turn, who do they talk to? And then lastly, our lab is really interested in the natural release of endogenous opioids like endorphins or enkephalin. So we want to know what are the potential sources of endogenous opioid modulation of the medial thalamus? So how could these endogenous opioids potentially inhibit nociceptive activity that's pathways that process through the medial thalamus? Okay, so we'll first start with, this is what I'm going to be talking about today during my talk. So we'll first start with the question of, is nociceptive stimulation uniform across medial thalamic subnuclei? But first, let's talk about measuring activity in the medial thalamus. So the previous studies showed these recordings from the different medial thalamic neurons one at a time. But we can actually measure activity using histology with the expression of an immediate early gene called CFOS, which peaks. This is a marker of neural activity, and it peaks, the protein peaks about 90 minutes after a particular stimulus, in this case, the stimulus of pain. So taking advantage of CFOS, we can capture CFOS expressing cells or pain or active cells with a technique called TRAP. We can capture these genetically. So TRAP stands for targeted recombination in active populations. So we can give a stimulus to permanently capture the permanently labeled CFOS positive populations. So with TRAP, you can capture all the ones that are active that within a 4-hour time period, and then label them in a color like red with TD tomato or green with GFP. Or in the same mouse even, we can give another pain stimulus and fix the tissue after 90 minutes to label FOS as well. So with the help of former Corder Lab PhD student, Dr. Lisa Marie Woldridge, and postdoc, Dr. Gregory Salamando, we first tackled this question of whether medial thalamic subnuclei respond uniformly to nociceptive stimuli with this first experiment in which we use this acute pain trap protocol. So in this acute pain trap, we give the mice 55 degrees Celsius hot water drops to trap the pain active cells. And then that causes a recombination and expression of a GFP in these nociceptive population. Then later, we give another stimulus later to look at FOS. So all the mice received this acute pain trap, but then the mice were broken into 4 different FOS groups. A control FOS, in which the mice remained in their home cage, or home away from home cage. Acute pain FOS, where the mice got the same exact 55 degrees Celsius hot water drops. A chronic pain FOS group, which is where we use a form of neuropathic pain, in which is a spared nerve injury to the sciatic nerve, which is kind of like a model of sciatica. And then a chronic pain plus light touch FOS group, where mice with this spared nerve injury received light touches to the hypersensitive part of their left hind paw to kind of like reactivate or sensitize these hypersensitive area. Okay. So then we did 3D volumetric analysis of these stimulus-activated medial thalamic cells with the help of a company called Life Canvas Technologies. So to do this, we first preserved the tissue and fixed it to preserve the protein structure and morphology, and then cleared the tissue with delipidation to reduce the scattering of light during fluorescent imaging later. After that, immunolabeling was performed to stain for these GFP from the trap proteins or FOS, and then the tissue was imaged with a light sheet microscope, which is a microscope that uses laser light to image three-dimensionally all throughout the brain, in this case, the medial thalamus, by imaging one Z-plane at a time across the entire brain structure. Then these images were registered to the Allen Brain Atlas, and we analyzed medial thalamus specifically, three-dimensionally. Note that we're looking at FOS to determine activity, which labels all the neurons that were activated 90 minutes after a particular stimulus, and TRAP to label the cells that were activated within the 4-hour time period. So before, what's happening? Okay, so before, I was showing you examples of studies that were doing in vivo or even in vitro, looking at activity from these cells on a millisecond or sub-millisecond timescale, but this is a much broader timescale. But we have the advantage of being able to look at activation in all the nuclei at once and compare, as well as looking at overlap between this trap and FOS to look at how different stimuli might affect the same cells. So we found that medial thalamic nuclei increase in response to acute and chronic pain. Here I'm showing representative images of FOS density in the medial thalamic nuclei in our 4 FOS stim groups. This no stim group with the home cage conditions, the 55 degrees Celsius hot water group, this nerve injury group that were also in this home cage, and then this nerve injury group that got that light touch to their injured hind paw. When we normalized FOS density to each nucleus in the uninjured home cage condition, when we normalized FOS density in each nucleus to the uninjured control conditions, we see that acute pain activates some midline nuclei like the IAM and XI on both hemispheres, as well as ipsilateral reunions, IMD, and CM. And you can see these here. Allodynia also activates many of these midline nuclei across both hemispheres, this chronic pain plus light touch group, with the exception of the PV, which is only on the contralateral hemisphere, the right side in this case. While chronic pain alone, this mice that had the nerve injury but remained in their home cage seems to trend towards a decrease in activity compared to the controls, with a significant decrease only in the ipsilateral CL. So if we want to, let's take a step back. If we want to target these cells to help relieve pain, we have to make sure that the same cells that activate to a pain stimulus activate again later. So, and these are making sure that they're stably involved in the experience of pain, right? If there's representational drift in which populations shift over time, it's harder to pinpoint exactly what cells to target. So we then asked the question of whether the cells that were activated during the initial acute pain trap were reactivated again during another instance of acute pain, or even reactivated again during the chronic pain plus light touch. To do that, we calculated the percent re-recruitment of acute pain trap neurons. So this is, of all the acute pain trap neurons, how many were re-recruited later during acute pain again, or re-recruited later during this chronic pain plus light touch. And we found that several of these nuclei were stably active across acute and chronic pain, at least with our FOS experiments. For example, things like the right IMD and the left RE were stably active across acute pain. In this case, there are significantly more acute pain trapped neurons that were expressing acute pain FOS than those that were just expressing control FOS, representing this population that is activated during both, that is significantly outnumbering those that are just re-expressed later at rest. And when we looked at those that were stably active across acute and chronic nociception, we found that the contralateral CL and the ipsilateral RE had significantly more neurons that were recruited during chronic pain plus light touch than those that were just re-recruited at rest. So these populations are stably active across acute and chronic nociception. So very cool. That previous study looked at the medial thalamic nuclei as whole 3D structures. And we've even established this atlas of activation within the medial thalamus that can be used for future studies for anyone who wants to look at these neurons in vivo or anything like that. But we also wondered whether pain phos activation in these nuclei occurred across the whole nucleus or even just the anterior or just the posterior ends of the nuclei. So next, we did a 2D slice-by-slice analysis to expand our atlas even more to look at stimulus-activated medial thalamic neurons at 4 different coordinates in the medial thalamus: negative 1.07, negative 1.31, negative 1.55, and negative 1.79 millimeters from bregma. So we repeated a very similar experimental design in which we had a acute pain or a trap mouse. This one is going to express TD tomato and pain active neurons. And we have our acute pain trap group as well as a home cage trap group. And then later, the mice were given 4 different phos stimuli, right? A home cage control group, an acute pain group, this time a light touch group, and then a chronic pain plus light touch group. This was done with a team of quarter lab mates. Appreciate you guys so much. Postdoc Raquel Adaya Sandoval Ortega, thank you. Malika Mahmood, who left and is now at Pitt for her PhD, and a fabulous team of Penn undergraduates. So first, we looked at just the effect of acute pain and compared the control trap to the acute pain trap group and found increased trap density in the acute pain conditions in both hemispheres of the CL, but only the anterior CL, as well as the MD, but only anterior MD, as well as the posterior part of the ipsilateral CM. Next, we looked at phos activation across our 4 phos groups: control, acute pain, light touch, and chronic pain plus light touch, and found that some nuclei, like the ipsilateral CL and ipsilateral PV, have increased activity to acute pain stim, but only in anterior sections of the nuclei, while ipsilateral IMD and MD and PC activate from acute or allodynius stimuli, but only in posterior slices. Several nuclei pop out from this atlas. You can look at a bunch of them, but note that the CL and the MD engagement here during acute and chronic pain. Just a little teaser. Teaching us that acute and chronic nociceptive activation in the medial thalamic subnuclei are certainly not uniform. And even within each nuclei, there are these spatial microdomains for nociception that differ even across acute and chronic pain, representing these acute and chronic nociceptive hotspots within and across medial thalamic nuclei. Next is to tackle the question of whether the mu-opioid receptors are expressed directly on our nociceptive or pain-active neurons in the medial thalamus. So, with the help of former Quarter Lab PhD student Jessica Wojcik and current postdoc Dr. Blake Kimmy, just both of them are doctors actually, we performed the acute pain trap protocol again. So this 55 degrees Celsius to capture these acute pain active neurons, and then did RNA scope or in situ hybridization to stain for acute pain trap and OPRM1 mRNA. This is the precursor to the protein. After that, the images were processed using an AI-assisted cell counter called Halo AI to quantify the mRNA in both of these probes. And here are some representative images of the expression of OPRM1 and acute pain trap in the medial thalamus, specifically here, the CL and MD thalamus. You can see there is a little bit of overlap here. Now let's quantify. So for OPRM1 specifically, we were interested in the number of OPRM1 transcripts that might be expressed per cell. So first, we quantified all 20,000 medial thalamic cells across all subnuclei in both hemispheres of 6 mice and plotted the distribution of number of OPR1 transcripts per cell, finding this non-normal skewed distribution of number of OPR1 transcripts. We found an average of 17.5 transcripts per cell, a median of 15, and information about number of transcripts in each quartile. And this is the representative images of some of these like punctate transcripts within the cells. We use these descriptive statistics about the number of transcripts to stratify the neurons into 4 different groups. So now we have like OPR1 expressing groups. First, we have this low expressors, medium, high, and very high expressing cells. And we found that 55 to 65% of cells that in this medial thalamus express both acute pain trap and OPR1, as well as so there's a very high percent of cells that express both. Additionally, 55 to 75% of the cells that are acute pain trap positive co-express OPR1. So 55 to 75% across the medial thalamus of the pain activated cells have this or potentially have this mu opioid receptor expressed. They definitely have the OPR1 mRNA, indicating that there is a heavy overlap between the acute pain active neurons and the OPR and the more positive cells, and giving evidence that the nociceptive medial thalamus might be a potential target for opioid-mediated analgesia, telling us that the potential for inhibition of nociceptive processing in the medial thalamus can happen in these pain-activated cells using more ligands. So knowing that, we moved in a different direction to look at the connectivity of the medial thalamic Moore cells. So the medial thalamus in general is very highly connected. It's connected to areas that are responsible for a variety of things in cognition and behavior. And there are some cell type specific inputs. For example, the PBN has TAC-R1 positive inputs to the medial thalamus. The PAG, this was done here by Blake Kimmy in the Quarter Lab, has more positive inputs to the medial thalamus. This is shown in green, as well as the spinal cord sending inputs to the medial thalamus. But the actual architecture of the medial thalamic Moore cell type remains unclear. So we wanted to map that out as well and add that to our atlas. And we started with who sends monosynaptic or direct inputs to the medial thalamic Moore cells in the medial thalamus. So for our medial thalamic Moore tracing studies, we had to choose a particular nucleus or a couple. So we choose to specifically target the MD and CL thalamus, CL and MD. This is supported by previous studies in the literature showing that the CL is necessary for effective pain responses. You saw this one earlier, that lesioning the MD thalamus reduces writhing responses in response to intraperitoneal acetic acid injection, as well as lesioning it reduces aversion to that pain stimulus as well. So we wanted to, also with our atlases, we saw that both of these nuclei activate in increased activity in terms of trap density and FOS density. This is not shown here, but you saw earlier that these nuclei were stably active during acute and chronic pain. And also former lab member, Dr. Nora McCall in the Quarter Lab did pain trap, same acute pain trap, and found that axons from the anterior cingulate cortex projected directly, especially the nociceptive ones, projected directly to MD and CL thalamus. So we wanted to hone in on these specifically to look at first the monosynaptic direct inputs to CLMD Moore neurons. To do that, we used the rabies tracing method. So aside from being one of the deadliest viruses in the world, rabies virus can actually be useful for neuroscience. This is a useful tracing tool because it can hop neural synapses. So we use a modified rabies virus strategy where we injected a rabies virus and Cre-dependent helper viruses into our target cells of interest, medial CLMD Moore neurons. And this was done in an OPR1 Cre mouse to get that cell type specific connectivity. And the OPR1 Cre mouse was developed by my committee chair's lab, Dr. Julie Blendy. Thank you so much for that. And here are the starter cells zoomed in into the CLMD here. we see widespread direct inputs to medial thalamic Moore cells, with the largest percent of total inputs coming from the habenula, the reticular thalamus, and the superior colliculus. And then here I have this visual representation, a map showing the weighted inputs across our top 22 input regions across the whole brain. Here are some representative images of inputs to 5 specific areas in the brain: the anterior cingulate cortex and the PBN, periaqueductal to gray, and the PBN. These are regions that are heavily involved in nociceptive processing, as well as the reticular thalamus and the reticular formation, regions that are known to regulate medial thalamic function. And we first wondered, is there a lateralization of inputs? So is there a hemisphere, left or right, bias of inputs from each of these regions? And we found that, for example, in the ACC, the ipsilateral, this is the same side of our injection, inputs greatly outnumber the contralateral ones in several layers of the ACC. Here you see it in the layer 5A, 5B, and 6A. This supports evidence in the literature that the anterior cingulate cortex sends layer 5 and 6 projections to this higher order medial thalamus for the function of retaining these recurrent thalamocortical loops that help keep the cortex updated about incoming sensory information that comes to the thalamus. And then in the context of pain, we think that it even might help maintain sustained attention to these pain stimuli. Okay. Next, we wanted to see if there was any sub-region specific biases from many areas. So we looked at the periaqueductal gray and found that sub-regions of the ipsilateral PAG, for example, the VLPAG, projects to the medial thalamus, CLMD more than others. Here we have VLPAG projecting significantly more than lateral or dorsolateral PAG. Side note, these two regions connect very well to work that Greg mentioned earlier that I've done in my PhD, participating in these other lab projects that are characterizing each areas, the anterior cingulate and the PAG, their activity during acute and chronic pain, and their involvement in the opioid system and the affective component of pain, working towards building this larger pain affective mu receptor network in the brain. And the PAG connectivity specifically with medial thalamic Moore is interesting because PAG is also involved in processing pain aversion, helping send signals, sending pain signals to the medial thalamus and coordinating defensive behaviors. So these areas connected with the medial thalamus help support what seems to be this network that works together to process pain and then coordinate pain behaviors. So first, we looked at the inputs to the medial thalamic Moore. Now we're going to look at the outputs from these CLMD Moore neurons. We did this by using the same OPERM1 Cree mouse, and we injected an anterograde virus this time, an anterograde Cree-dependent M-cherry virus into CLMD. This project was aided by Penn undergrad Ala El Hareka, thank you so much, and postdoc Raquel, who developed Axoden, our axon density analysis software. So we looked at the top 25 output regions from CLMD Moore neurons and quantified the percent of each area that are covered by projections, and finding that the greatest amount came to regions like peduculotegmental nuclei, dorsal raphe, central gray, and zona incerta. Then we wanted to quantify the percent of the total projections from the CLMD Moore neurons throughout the brain to our top 25 regions. And we saw heavy outputs going to areas like a lot of them in the frontal lobe, like the anterior cingulate orbitofrontal cortex, as well as the areas like the dorsal striatum and the nucleus accumbens. So now we've investigated the overall circuit architecture of CLMD Moore neurons, and we see a connectivity profile that is very broadly distributed across the brain, but is selectively biased towards circuits that are involved in affective component of pain, salience, wakefulness regulation, and overall behavioral coordination. So our lab is next interested in endogenous specific opioid inputs to the medial thalamus. We're interested in endogenous opioids because unlike exogenous opioid drugs, these naturally released endogenous opioids are released in spatio-temporally controlled manner to provide analgesia by inhibiting directly onto nociceptive circuits. And the release does not cause the harmful side effects associated with exogenous opioid drugs, right? You have mu opioid receptors all over your body, all over your brain, in reward areas, even in the areas of your lungs that help control muscle contraction for breathing. So when you give opioids to the whole body, that can have all these outside side effects, but this is very spatiotemporally controlled and direct, providing a potential target for safer and more effective analgesic or pain relieving therapies. And then endogenous opioids are released during things like pain expectation and acute stress. And then I could give another hour-long talk about how I modeled pain expectation in mice during my PhD, but this is not included today. But the most commonly known endogenous opioid are endorphins. Also, we have enkephalins and dynorphins, but today I'm going to be focusing on enkephalin. So what are the potential sources of enkephalinergic modulation of the nociceptive medial thalamus? Could these block these ascending nociceptive transmission at the level of the medial thalamus? So first we thought, what if the source of enkephalin to the medial thalamus was local, happening within the medial thalamus? Though we see a great amount of enkephalin signal within the medial thalamus, this is the IHC I did here in the lab, the in situ hybridization data, this one from the Allen Institute, shows that there are little to no enkephalinergic somas within the medial thalamus. So it looks like the enkephalinergic modulation of this population will be coming from long-range inputs from different parts of the brain. So we wanted to hunt to find these long-range inputs. To investigate enkephalinergic inputs from throughout the brain to CLMD neurons, we used a retro color flipper virus. This, in the presence of Cree, here we have the enkephalin pank Cree mouse. In the presence of Cree, the virus is going to express GFP in enkephalinergic, enkephalin positive inputs to the medial thalamus. And then in the absence of Cree, the inputs to the thalamus that are non-enkephalinergic are going to express M-cherry, or in this case, it's going to be blue. So we can look at enkephalinergic and non-enkephalinergic inputs in the same exact tissue to the medial thalamus, or to, in this case, the CLMD. Then we took it one step further. We also did another acute pain FOS stimulus in these mice. So we gave them this 55 degrees Celsius hot water drop. 90 minutes later, looked at FOS to, in the same tissue, look at potential nociceptive inputs to the medial thalamus, the enkephalinergic ones, and then the non-enkephalinergic ones. Okay. These are representative images from our top or most dense input regions: the habenula, the anterior cingulate cortex, the retrosplenial cortex, the perioccipital gray, and the parabrachial nucleus. Notably, I think this is really cool, we found that the endogenous opioid inputs in the terms of enkephalin to the medial thalamus are distinct from the nociceptive activated inputs to the medial thalamus. So when we calculated the percent of enkephalinergic inputs that were also pain active, and the percent of non-enkephalinergic inputs that were pain active, we found a greater percent of non-enkephalinergic inputs versus enkephalinergic inputs that are pain active, especially in the ipsilateral habenula, PAG, and PBN, suggesting that these opioidergic inputs to the medial thalamic soma from these areas are largely distinct from nociceptive inputs, and that these opioidergic versus nociceptive pathways to the thalamus operate in parallel and don't come from the same exact cells largely, and that these follow separate pathways. So, several conclusions can be made from all the work I showed you today. Acute and chronic nociception activates non-uniform bilateral hotspots across the anterior-posterior axis of the medial thalamus. There are distinct acute nociceptive medial thalamic neurons that are reactivated during subsequent instances of acute nociception and during mechanical allodynia. Most of the nociceptive activated neurons in the medial thalamus co-express OPRM1. Endogenous opioid afferents to the medial thalamus arise from largely non-nociceptive activated populations, giving evidence that the medial thalamic Moore population might be ideally positioned for circuit-targeted gene therapy modulation of pain or nociception. So many things can be done to move this work forward, right? I've established this pain and opioid atlas, as well as the inputs and outputs from these medial thalamic motor neurons, but there's work to be done to really determine whether this is a good target for pain relief. And the first is to, in vivo, in living mice, manipulate functionally the medial thalamic motor population. So would inhibiting the medial thalamic motor neurons reduce pain-related behaviors in mice? Potentially would inducing encephalinergic transmission onto medial thalamus reduce acute and or chronic pain-related behaviors? We're also interested in investigating further the stability of this nociceptive neurons across multiple types of pain or across pain states. So we want to do longitudinal monitoring of medial thalamic motor neurons activities in vivo across the acute to chronic pain transition using something like miniature endoscope technology. And then our lab has this cool tool. It's called Lupe. It's a light automated pain evaluator to detect spontaneous pain behaviors after nerve injury. So the question is, does manipulation of medial thalamic motor neurons affect these non-evoked or spontaneous pain that might occur? So that being said, thank you so much for listening today. I would like to thank my lab, appreciate you so much, Quarter Lab, as well as my funding sources. There's a variety of them that help fund the Quarter Lab. Specifically, like Greg mentioned, the DSPAN, which has funded the last 2 years of my PhD and will provide up to 4 years of funding for my postdoc from the NIH. I'm so happy that I got to share all of this with you all, and I'm happy to answer any questions you might have now. I'm going to take questions in person as well as some questions from the Zoom webinar as well.
Great job.
Thank you.
Activated by negatively. I'm assuming. Is that true?
Yeah, I'm sure I've read that the lateral habitula is activated to painful stimuli.
Okay, so thalamus.
Yes, encephalinergic.
So, um, you think that specifically part nociceptive, but
Do I think that the projection specifically from the question is, do I think that the projection specifically from the lateral habitula, which is pain activated, to the MDCL thalamus could be manipulated specifically?
yeah, exactly. Like something.
Yeah, I think that one could perform that experiment looking specifically at, you probably have to use double intersectional methods. So you have the mu opioid receptor population. You could do a projection specific population as well, and then specifically manipulate that one, inhibit it or activate it to see how that affects pain in vivo. The thing that me and Greg, to our greatest frustration over the course of the PhD, is that the habitula is directly above the medial thalamus. So there would have to be some pretty sophisticated techniques to manipulate the habitula without getting any of the virus that you're using into the medial thalamus and vice versa as well. Any other questions in person? Go on. Oh, interesting. So you're asking, are enkephalnergic inputs to the medial thalamus? Or is there any in vivo like evidence of how those particular inputs act during pain? Actually, nobody has gotten there yet. I don't think I've seen studies of, I've only seen anatomical tracing studies of enkephalin into the medial thalamus. Now, there are in vivo studies, for example, of how enkephalin or endogenous opioids or opioids might modulate the activity of mu opioid receptor positive cells in the medial thalamus, but not at their soma, at their axons. So there's an in vivo study that looked at the projections from the medial thalamus, MD specifically, to the striatum, and specifically the mu opioid receptors that are on the axons in the striatum. And they found that in vivo, microinjecting Damgo there, specifically onto those medial phalamic Moore projections, reduces, actually, this is an in slice, I'm sorry. It reduces activity, pain activity there, but I actually don't think anyone's looked at in vivo, does that then reduce pain activity? In vivo study in the medial. I think that there's an in vivo study from the MD to the ACC, and that was done to also inject mu receptor ligands at the level of the ACC from the MD thalamus. And I think they found a reduction in pain aversion. But they used, I'm pretty sure they used Damgo, which is a modified enkephalin, but it's not the actual enkephalin that comes from this like natural release that might occur from pain expectation or stress. So that would be an interesting experiment to do and look at a particular projection too. Any other questions? Yes, Rakshita? Thank you. Manipulation. Interrupt their function. Okay. In my understanding and of reading, so the question was, would lesioning, specifically medial thalamus, relieve pain, but it would also affect your general experience of touch? In my experience, I don't think that those studies reported that. I think the people were still able to experience touch. And I'm not exactly sure about this, but I think that lesioning lateral thalamic nuclei I think it would affect your touch as well. But then, of course, like would also increase your pain. So, other questions? Hmm? Yes? Medial thalamus or CLMD, yes.
So, but negative valence, like so if that are encoding pain are these inputs need to be exploited if it isn't happening.
Okay.
Does that make more sense?
It does make sense. I think you said like, why would you need to experimentally induce enkephalin release from these input areas to relieve pain? Wouldn't these areas just naturally relieve pain in these endogenous conditions in response to things like pain, expectation, or stress? I think the answer would be, first, let's see if in natural conditions, do these areas release enkephalin during pain, expectation, and stress? It's generally known that endogenous opioids help to relieve pain, but they act very short. So, for example, you might be in the context of placebo pain relief. You might be, not everybody's a placebo responder. Not everybody is stably a placebo responder, so they might not relieve their own pain the same exact way. Patients that live with chronic pain also have deficits in central modulation of their pain. So I say all that to say, we want to kind of eventually provide a more reliable way of targeting them, because if we could all think really hard about relieving our pain, I'm sure many of us would not be here today. We're trying to figure out how to solve it. Okay, I'm gonna look at the Q&A. Okay, I got a question of how do you evaluate the pain in mice since they don't speak? That's a good question. I didn't talk a lot about our pain behaviors. And does mice behavior outside the experimental setup or those recorded too? And how did you evaluate those results? So when we did our pain mapping, we did not specifically record pain behaviors, but in the lab, we do many of these same acute hot water stimuli, chronic pain like SNI, nerve injury models, light touch, and we do see in the lab that, and I've done it specifically, those mice have a heightened sensitivity to non-painful touch. How do we measure their exact pain stimuli? So imagine you put a hot water on a mouse's paw. It can engage in reflexive behaviors, withdrawal behaviors. It will pull its paw back. It will do things like guard its paw, so hold it up. Like, what are you doing? I don't want you to be able to touch that. By the way, this hot water is not something that can permanently damage the mouse. This is just like something that they don't. It's hot enough for them to not enjoy it, but not anything like that will like eviscerate their skin or anything. So they can withdraw. They sometimes will jump, try to escape these motivational behaviors, and things like that. Hope that answers your question. I got the question of whether my animals were males or females. It depends on the study. For the anatomical tracing studies, for the 3D ones, those were all male. No. Those were a mix of males and females. They were half and half. For all of the pain FOS mapping, 3D and slice by slice. For the opioid receptor atlas, those were also half male, half female mice. And the encephalinergic projections were all male mice. I have a question that says, my question is about the endogenous opioid regions of the brain that you identified that projects the medial thalamus for pain. Thank you, Alana, for this question. Do those regions you identified have overlapping functions known in other processes or behaviors, like memory or hunger, for example? I guess it depends on the region. Let's look. Okay, so the retrosplenial cortex is important for the keeping how do I say this? Our internal sense of self and spatial learning. The periaqueductal gray is important for pain expectation. Oh, you said outside of pain. Outside of pain, the periaqueductal gray is important for defensive and aversive behaviors, for things that are not painful but are still not pleasant. I hope that answers your question. Thank you so much. Do I have any other questions in the audience? Shauna? The question would be, for those that want a PhD, what would be? Okay, the first question is, how do we select? Was there a selection process for our mice? We used adult mice, so those are over 50 days old, and we try to do a mixture of both sexes. And then we don't use mice that have any like clear disabilities or anything that like might confound the studies. We do not use singly housed mice either because loneliness can impact stress and things like that. And my answer to the question, the second question was, what advice would I give for somebody who wants to pursue a PhD, even within this field or without? I would say, go for it, identify your interests, talk to mentors. You can even reach out to me on Instagram if you would like. I say, if you think that you have questions that you're really interested in, problems that you really want to solve, and you have the support in your life to make that happen, go for it. You can do it. Don't think that you're not smart enough, because this isn't really a game for geniuses. It's a game for those who are determined. So I would say, go for it. Okay, thank you all so much for your questions. We're going to, so this concludes the public defense, and then we'll ask everyone to leave the room, and then I'll have a 1 hour long private defense with my lovely committee. Don't leave. I'm sorry. I forgot the acknowledgements. Wait, let me acknowledge my people, y'all. Hold on. I understand if you like have to go, but yeah. So first, I want to acknowledge my lovely family. Thank you so much, everybody in my family. For your support throughout the years. I do not take it, I do not take for granted all the love and support that you pour into me. I do not hold resentment for how often you ask me when I'm graduating. I really appreciate you. Thank you for providing this beautiful community to enrich me culturally and emotionally. I really love you guys. Those that are here in the US, those that are back home in Cameroon watching, thank you so much. I really do love you guys, and I thank God for you in my life every single day. Especially Auntie Estella, who couldn't be here today, but she's my academic hero, my auntie. Thank you so much, Auntie Estella. I'm gonna keep going, especially my mom. This is my mom. I appreciate you, mom. Thank you for all the love. I've called her crying ever since college. Go ahead again. That's my mommy. Thank you so much, mom. Thank you for your support every single day. Thank you for being so proud of me. Thank you for bragging about me to all your friends. And okay, my cousins, my uncles, aunties. Thank you to my friends from Pitt. I appreciate you all for being friends with me even to this day. Some of you guys are here in the audience. Some of you guys came from far away from Boston. Some of you guys flew in 10 minutes before the talk. Like, I really appreciate you being here and the role that you play in my life. Thank you to my friends that I've made here at Penn. Through BGAPSA, through EE Just, through organizations that help to support students like myself. You guys made a huge difference in my experience here. And though we started during COVID, we still got nice and close, and we still continue to support each other and lift each other up. And without your support, I would not be here either. Thank you so much. And thank you to my home friends. Thank you to Marlene and Shirley. And these are just some like miscellaneous pictures as well from a bunch of people because I couldn't fit them on the other slides. Thank you to Michael, who I met recently, but has made a huge difference in my life. Love you so much. Thank you so much for being a part of this experience and for supporting me. And a special shout out to two family members that we have. Why are you doing this? Two family members that unfortunately we lost this past year. My grandma, my mom's mom, Grandma Betty, and then my uncle Ojuku, who unfortunately is another victim of the fatal opioid crisis. We love you guys so much. I know you're watching from above, and I'm so sad that you couldn't be here with us today, but we really do love you, and you are in here with us, and we really, I just want to thank them specifically too. I mean, my grandma is the one who's the reason that all of us are here in the US today. And I came to the US when I was 4, and this is a product of the work of all the people that came behind me. And now they're all looking at me either from above or here on this earthly plane. So proud. So I am so happy. Thank you so much. Thank you so much for those who joined on Zoom. If you're watching and you supported me online, I really appreciate you as well. And those who support me online that are here in person, I also appreciate you. Okay. Yes, this room is booked. Okay, great. Okay, when you go out, you know, for sure.